My name is Marie Csete, and I'm a visiting associate in medical engineering at Caltech and professor in the USC anesthesiology department, concentrating on basic research, and most importantly, an independent consultant in regenerative therapies. This tutorial about lessons that can be learned from Alzheimer's disease for AMD and vice versa comes about because of many of the common features of the two disease processes. Whether macular degeneration is Alzheimer's disease of the eye is something that I'll leave you all to ponder when we talk about the evidence for A beta driving macular degeneration. But one thing we can say for sure is that because the eye and the brain are developmentally from the same source, the patterns of cellular senescence will be similar in the two organ systems. And so the lawn on which degenerative processes occur is very similar in the two organs and may well explain some of the similar pathologic responses in the two degenerative diseases. So in addition to the common developmental origin, the two diseases share similar risk factors. The usual things that your cardiologist will tell you to control are important in delaying both Alzheimer's and macular degeneration. Age is the major risk factor in both diseases, and both have a very similar female predominance. Again, the inflammatory environment of a healthy eye and a healthy brain are relatively similar compared to other organ systems because of the blood tissue barriers. Both pathologies are characterized by oxidative stress, inflammation, mitochondrial dysfunction, lipid and glucose metabolism dysregulation, probably vascular hypoperfusion early in disease. And the one striking similarity in pathology are the extracellular plaques versus drusen that seem to drive the pathology. An unfortunately similar common feature is that both diseases are poorly reflected in animal models, and there is insufficient clinical phenotyping that has dogged clinical studies in both diseases, something that I know this group is taking on for macular degeneration. So here are the pathologic features in common: drusen and plaque. Plaque is generally considered to be A beta driven. The neurofibrillary tangles are intracellular and driven more by the tau protein accumulating. Important differences between the two diseases are something we'll highlight during the course of this tutorial, and they're important to learn from as well. So the genetic susceptibility genes are largely non-overlapping, and I'll say something about ApoE4, which is the most important genetic susceptibility gene for Alzheimer's later in this talk. For early onset Alzheimer's disease, another way of thinking about genetic susceptibility for Alzheimer's, APP and the presenilin mutations are genes that deal with lipid processing. They only account for a minority of cases. We really don't know the vast majority of early onset Alzheimer's causes. ABCA4 for Stargardt's will come up a little bit later, since ABCA1 may be involved in Alzheimer's pathology. The mitochondrial haplotypes associated with Alzheimer's and AMD are completely different, which is interesting. And again, unfortunately, a common feature is that failure of drugs targeting these gene products under control of susceptibility genes have largely not led to potent therapies for either disease. The Alzheimer's community has been wringing its hands over this, and there's a general consensus that drugs targeting A beta efflux are being delivered too late in the disease process. Perhaps targeting only one molecule will never work in these complex pathologies. Perhaps we haven't identified the upstream drivers. And again, insufficient phenotyping of patients. AD is very heterogeneous, as is macular degeneration. So starting with the common epidemiology, age is the major risk factor. Alzheimer's disease is more common, unfortunately, than macular degeneration, with a 10% prevalence in people over age 65. However, the race distribution is very different between the two diseases, with African Americans twice as likely as whites to get Alzheimer's disease, and Hispanics 1.5 times more likely than whites to get Alzheimer's. Some of this may be due to hypertension in the African American community and less control of cardiac risk factors. There's a striking female predominance of both diseases, and it's about the same. 65% of patients with Alzheimer's, like AMD, are women. In the Alzheimer's community, this is just generally attributed to longer lifespan in women, but I think Robbie Brinton's take on this is pretty interesting. And she's shown that there's the highest risk of development is in those with the risk factor ApoE4 alleles and certain mitochondrial risk haplotypes, which turn out to be driven by gender. And the way the Alzheimer's disease community puts this all together, thinking about the mitochondrial haplotype, is shown on the left of this slide. And the way the macular degeneration community with Chris Kenny's work thinks about mitochondrial dysfunction is a little bit different. And what you'll see is that the mitochondrial haplotype focuses on things with macular degeneration like complement that are well known to be involved in the pathology, and similarly, the mitochondrial haplotype in Alzheimer's disease focuses on A beta production and plaque deposition. But are they talking about the same things? We'll take a stab at that a little bit later. It is really different in terms of mitochondrial haplotype. In fact, it's opposite. So the haplotypes that mediate decreased risk in Alzheimer's disease are actually the haplotypes associated with AMD. The JTU haplotypes are associated with AD. And for Alzheimer's disease, the H haplotype and HV superhaplotype are associated with risk for Alzheimer's disease, and they're definitely more risk if you are a woman. Nonetheless, I think that as methods to preserve mitochondrial function or improve mitochondrial function come online, they may yield benefits for both Alzheimer's disease and macular degeneration. And thinking ahead a little bit, I'll give 2 examples here of things I think both communities should be and maybe are thinking about. Allopregnanolone increases neuroprogenitor proliferation in AD mouse models and reverses the cognitive defect, and there's a graph of that on the right. This work served as the basis of an ongoing clinical trial. Now, allopregnanolone is also a potent inducer of mitochondrial proliferation. Humanin is a mitochondrial-derived peptide, which has been shown in various preclinical models to be broadly neuroprotective, including against direct A beta toxicity. And there's also work suggesting that humanin is protective of ER stress in cultured RPE. The only clinical trial I could find in clinicaltrials.gov, however, has to do with cardiac disease. Humanin is also a mitochondrial proliferation inducer. So this overlap gets us back to thinking about ApoE4, and it is the major risk factor for AD. And as you can see, if you are unfortunate enough to inherit 2 alleles of ApoE4, you're virtually guaranteed to get Alzheimer's disease, and you'll also get it at a younger age. So I'm not going to go into detail at all about A beta metabolism. It's a very complicated system, but these various graphs and figures that I'll show you and references are there for your future reference if you need them. The main A beta clearance pathways are receptor mediated in neurons and in glia, or A beta can go into the interstitial fluid or through the blood-brain barrier into the spinal fluid and be processed that way, as well as undergoing proteolytic degradation. When there's impaired clearance, which happens, as you will see, in Alzheimer's disease well before the onset of clinical symptoms, A beta accumulates in the brain. The neurotoxic oligomers can be cleaved off, forming amyloid plaques. And there is also perivascular accumulation of amyloid beta, and cerebral amyloid angiopathy is a quite common co-traveler with Alzheimer's disease. So ApoE4 is sort of a central player around A beta processing that contributes to the pathology of Alzheimer's disease, but also has effects on things that you think about in terms of macular degeneration, glucose metabolism, neurogenesis, vascular function, mitochondrial function, etc. So the common pathology really comes down to these extracellular accumulations, lipid and protein in plaques and drusen. So here's a list of some, probably not all, that are the molecules found in both. I've highlighted A beta, cholesterol, and PLA2 for a little bit more detail because I think that these are things that may be of particular interest to the macular degeneration community. First, we'll start with cholesterol. The brain synthesizes its own cholesterol, the glia synthesize cholesterol, and completely exclude circulating cholesterol from the brain via the blood-brain barrier. Also, the brain cholesterol is different than liver synthesized cholesterol. It's got an extremely long half-life in the brain. Retinal cholesterol is also synthesized locally, but also taken up from the circulation. Unlike the blood-brain barrier, the retinal blood-brain barrier is permeable to lipoproteins that transport cholesterol and other lipids. In the retina, there's a lot of cholesterol turnover with outer segment turnover. And through Christine Curcio's work, we know that cholesterol and cholesterol esters accumulate in retina with normal aging and with AMD. Interesting work at USC and at HMRI just recently showed that cholesterol efflux is impaired early in Alzheimer's disease at the MCI stage, the mild cognitive impairment stage. This study focused on the ABCA1 mediated cholesterol efflux, and there is a loss of function of ABCA1 model of Alzheimer's disease. So this efflux problem happens early in disease. Efflux of A beta precedes this, but the investigators didn't look in preclinical Alzheimer's disease patients, so we don't really know how early this cholesterol efflux happens. So it raises a new potential target for therapy that may be relevant in AMD. Cholesterol efflux is important because when the membrane content of cholesterol is reduced, it favors formation of A beta peptides. and the reduced neuronal membrane cholesterol favors production of non-amyloidogenic A beta peptides. Increased neuronal membrane cholesterol, so when efflux is messed up, increases presenilin and base, favoring the amyloidogenic A beta peptides. Drugs that increase cholesterol efflux include a retinoic acid X receptor agonist, bexaroxetine, which is in clinical trial for AD. These RXRs are also protective against oxidative stress in cultured RPE lines. So whether, again, common target becomes important, this is something to think about. But if you really want to know about cholesterol in the retina and in AMD, you should ask Christine Curcio, and I'll be very curious to hear what her take on the AD literature is. So the picture here is from one of Christine's papers showing the location of cholesterol in human Brooks membrane and in drusen. Is the perivascular accumulation of lipid in the retina similar to that which is happening with cerebral amyloid angiopathy? I don't really know. But I think there is a reasonable question to ask that will clearing cholesterol at some early stage help to reduce drusen and plaque formation, or in later stages help to reduce drusen and plaque burden and therefore disease severity. So A beta is really the big one to think about in terms of the relationship between Alzheimer's and AMD. So as I mentioned before, if you have evidence of A beta reduced efflux into spinal fluid, that is highly predictive of developing Alzheimer's disease in the future. So in the spinal fluid, you can tell people with pretty much 90% certainty that they will develop the disease if they have a reduced A beta tau ratio in their spinal fluid. The other way that risk for Alzheimer's is being addressed around A beta accumulation is now with PET imaging. And I attended the 2016 Clinical Trials for AD conference, where now that there's a lot of images of A beta PET in the literature, there is a general consensus that the A beta burden has to be considered for enrollment in clinical trials. There's a problem, however, in that A beta imaging alone does not correlate with the disease state, with the cognitive state. And I will propose that just as A beta accumulation in the brain doesn't give you definitive information about the cognitive state, A beta accumulation in the eye will also not correlate well with Alzheimer's disease state, but still may be useful with other modalities for diagnosing Alzheimer's disease. So again, the clearance of A beta peptides is pretty complicated. In the brain, they are phagocytosed by microglial populations and then undergo further degradation by insulin-degrading enzymes and nephrolysin. They also efflux through the LDL receptor called LRP at the blood-brain barrier. In the peripheral circulation, A beta accumulates outside the CNS, and we're learning more and more that A beta, when it's accumulating in brain in Alzheimer's, is accumulating all over the place. Vasculature, the heart, the pancreas, the liver, for sure. The peripheral circulation clearance involves the soluble LRP1 receptor and soluble RAGE receptors. So another issue in clearing A beta out of the brain and into the spinal fluid is that it has an important physiologic function. It has neuroprotective properties, it has neurogenic properties, it is an important modulator of synaptic plasticity. sequesters ions, antioxidant activity, certainly in maintaining the blood-brain barrier structural integrity, and the role in calcium homeostasis. Pathologically, it is at the center of Alzheimer's, and all of these toxic properties are listed on the left. They require not only high levels, but they require an extracellular location. If you could keep A beta inside the cell, it would be much less damaging. Of course, A beta in drusen is outside the cell already. And also the oligomeric form of A beta is very important. The A beta 1 to 42 is the most toxic form. And again, this is just a reference for you to have showing the various degradation pathways and processing pathways for A beta. So although A beta PET imaging has been a bit of a disappointment in terms of its relationship to cognitive decline, a recent study suggests that when you combine two PET images, one for A beta and one for tau, you can have a good imaging correlation with the cognitive status and with those CSF markers that I told you about, the reduced A beta to tau ratio. So there's not a whole lot of experience yet with tau PET, although I know it's available at USC, which has an Alzheimer's Disease Research Center. Both are expensive, but they give important compartment diagnostic information about the brain with a lot of mathematics involved. There's no information yet on how these signals will change with therapy, so if they'll be useful beyond diagnosis, and whether clearance of the combination of A beta and tau in the images will correlate with improvements in clinical severity with therapy. So back to A beta accumulation in the retina. It accumulates everywhere, basically, with age. In RPE, in Bruch's membrane, in outer segments, and choroidal vessels, and, as I said, in drusen. So what could that mean for macular degeneration? There's some evidence to suggest that A beta exposure in RPE will cause an upregulation of VEGF. There's other evidence to say that A beta may work by activating the alternate complement pathway or lowering the threshold for complement activation. But a recent study raises an interesting question about A beta as a driver of macular degeneration. In this study, A beta 142, which is the toxic oligomer, was injected in the subretinal space of mice. And very rapidly, after about 1 week, the retinas had features of age-related macular degeneration, including pigment abnormalities, disrupted hyperplastic RPE, disrupted Bruch's membrane, and loss of outer segments. What's going on here? Is this really a new model of AMD induced by A beta? Well, the injection puts the toxic AB directly into the extracellular space and is not preceded by accumulation of A beta in the intracellular compartment of the retina. But nonetheless, I think it's compelling model to look at what small levels of A beta extracellular may be doing in driving the parts of AMD pathology that are well recognized. The last molecule that accumulates both in drusen and in plaque that I wanted to focus on was phospholipase A2. This is also seen not only as published in increased levels in the spinal fluid of those with mild cognitive impairment, and then more so with late onset, full-blown Alzheimer's disease. But phospholipase A2 is also seen in higher levels in the spinal fluid of patients who are at risk by their spinal fluid A beta and tau ratio for AD versus those who are age-matched and not at risk by virtue of the spinal fluid A beta tau ratio. This study is also from HMRI, led by Alfred Fonte, and I think it emphasizes the importance of looking at both the soluble fraction of CSF and the nanoparticle fraction of CSF. In most of the studies that Alfred has done in Alzheimer's patients and pre-Alzheimer's patients looking at spinal fluid lipids, most of the actions in these nanoparticles that are, some of them are exosomes, but a lot of them by EM look to be shed membranes. So they reflect not only cell death in the brain, but they reflect the lipid oxidation changes that have happened over the course of disease. There's been very little work looking at the nanoparticle or exosome fraction in the fluid compartments of the eye, as far as I know, although Jerry Subag and the HMRI group are now starting to do some of that with vitreous samples. So the phospholipases regulate membrane lipid homeostasis, oxidative stress, inflammation. They're, again, one of these central pivotal players. And phospholipase A2 being elevated sets off a very nasty arachidonic acid inflammatory cascade that you see here. When you look at the lists of drusen content that are in review articles, you don't usually see phospholipase A2, but I was able to find 12 different reports that indeed phospholipase A2 is in drusen, or at least in some drusen. And so I think this is, again, a common target that we can think about between Alzheimer's and AMD around the arachidonic acid pathway. Alfred's work in spinal fluid in Alzheimer's disease has also showed that there's a specific reduction in acid sphingomyelinases and a variety of changes in sphingolipids that are really complicated, but again worth looking into because they raise potential common drug targets for the two diseases. And this is a surprising result that we got with iPS cells in my lab at HMRI, where we looked at patients who were not yet clinically compromised with Alzheimer's, but were either at risk or age-matched and not at risk by their spinal fluid A beta tau ratio. And surprisingly, before differentiating the cells, so in their fully undifferentiated state, those derived from normal cognitively healthy patients who were above age 70 versus cognitively healthy patients with risk factors for Alzheimer's disease underwent less apoptosis than the Alzheimer's at-risk patients, just at baseline. So what could possibly be going on here? We're sifting through data, and it's a bit confusing, but reprogramming to the iPS state rejuvenates the chromosomal genome. And presumably the chromosomal genomes, although we didn't sequence these things, were pretty much the same after reprogramming in these two populations. But there's very little information on how mitochondrial genomes are reprogrammed during iPS induction. And perhaps the reprogramming process is damaging to mitochondria, and the Alzheimer's disease patients are more at risk for this damage. Perhaps the plasma membranes that we start with from the T cells that were used for the reprogramming source are not repaired with reprogramming, and the increased apoptosis we're seeing is a result of all of these membrane changes that Alfred is seeing in the Alzheimer's membrane and spinal fluid. But it made me think and look back into the IPS literature from AMD patients, and almost everyone just goes straight to differentiating into the operative cells, and no one is looking at the baseline IPS characteristics as far as I can tell from AMD patients, and there may be information there before you actually go ahead and differentiate the cells. So I hope this has been a little bit provocative. When I set out to do this, I realized how little I knew about Alzheimer's and how little I knew about macular degeneration. But I think that we can first agree that the common developmental origins will mean that the senescence patterns laid down in the brain and eye set up for similar degenerative processes. The similarities in the molecular makeup and drusen and plaque are compelling and make it reasonable, I think, for ophthalmologists to be hooked onto these big AD clinical trials, if possible, to be looking at the effect of clearing A beta, when we can actually do that, or other disease-modifying drugs on the eye. But differences in the molecular makeup between drusen and Alzheimer's are also going to be informative. And if I had to make my best guess about molecules in drusen that may be informative for developing therapies, I would say phospholipase A2, the arachidonic acid pathway, is something to think about. And on the other side, although you can find that complement is present in plaque, it's been very, very poorly studied in Alzheimer's, and I think deserves another look. Obviously, future common targets around mitochondrial health hopefully will yield some therapies for both. In terms of diagnosis of Alzheimer's, I think A beta presence alone in the eye will not likely be useful, because it's not useful in the brain. But in combination with other eye imaging techniques, it may well be useful, and certainly Alfredo Sadun has made a good case for Alzheimer's disease being accompanied by retinal ganglionic cell degeneration. And other people have thought about this now, that maybe multimodal eye imaging holds promise for AD diagnosis, and certainly Alfredo's work should be in this, which it isn't. So I think the lesson for Alzheimer's and AMD, if you look through this painful literature of the common molecular players, is happening all throughout medicine, that just as in the diagnostics we discussed, the therapies will inevitably have to focus on more than one target. But for AD, I think looking beyond drug targets is becoming increasingly important. So exosomes, again, that are complex therapies are something that I see happening in this in the preclinical space. And importantly, neuromodulation to move lipids around and to change the molecular conformation of lipids from toxic to non-toxic forms in preclinical studies is very encouraging. And I think as a way to encourage efflux independent of receptors, neuromodulation may be important in Alzheimer's disease. I don't know how that would play out in AMD. And I would encourage people who are making iPS cells from AMD subjects to look at the undifferentiated state of the cells for clues to the disease pathology. Thank you, and I hope for an interesting discussion at this year's meeting.